Okay, next up we have Mary Lou.
Okay, so I wasn't supposed to talk. Marllon was supposed to talk, but he is very ill this morning. And so I will do my best to give his presentations. We have two short presentations because he basically has two projects going on here in Ventura County. So the first one I'm going to talk about is basically he's titled Biology Tells Us What Could Work and Data Tells Us When It Actually Does, because Marllon is a fanatic about data and data analysis. He has a PhD in horticulture, but he also has a master's in big data management. So this is sort of repeating some of the slides that Anaki gave, but just to remind you that pollination is the transfer of pollen from the anther to the stigma by some agent. We've heard all about that from Hamutal and from Anaki. And fertilization is the process in which the male gamete from the pollen grows down and fertilizes the ovule and forms the zygote. And so these are just some pictures of, does the pointer work? No, no pointer. You can see the pollen, whoops. Yeah, you can see the pollen, on the far right, you see the pollen grains on the stigma with growing down, and on the right next to it on the right are the pollen tubes, and this is actually flowers from Ventura County. So the process of pollination, as Anaki talked about, pollen grains need to be deposited on the stigma. The stigma needs to be receptive, which Anaki talked about. It's the female phase. And then the third major step is you need to get those pollen grains to grow down to the ovule, and one of them will win the race and fertilize the zygote, the ovule. So this is older data before that we collected at the Devonshire farm in near Oxnard, and we had multiple varieties in this block. We were looking, and Anaki shows some of the data, but this just shows in 2002 that you can get overlap, but every variety has a little bit of a different window of time that they want to flower. And this slide is data from Irvine, again collected many years ago, but it highlights the variability you have from year to year. So if we focus in on 2004 and 2005, the green and the red bar, you can see that in 2004 we had higher air temperatures, lower rainfall, lower humidity, and the bloom duration, especially like in Lamb Hatch, was very, very short. Okay, you compare that to 2005, where we had lower temperatures early in the flower season, we had higher rainfall, higher humidity, and we had a much longer flowering season. So I'm going to talk about first the California Avocado Commission funded project. You can see is myself, Anaki, and Marllon. And Marllon is the person doing all the work, and Anaki and I are sitting back telling him what to do, adding more work for him to do. And if Marllon was here, he would say, yes, 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 because we piled on a lot more work yesterday. So we have 7 sites here in Ventura County. We have Moore Park, Fillmore, Soares, 3 sites near Santa Paula, 1 to the east and 2 to the west. And we have 2 of the sites where we have gem. So that's it. And we have 1 year of data. And we went out and we had him collect all kinds of data, flower density, pollen germination, flower viability, et cetera, and he set up a little mobile lab in an apartment, a room he rented here in Ventura, and collected all kinds of data and took the flowers back to Riverside. And he has been very, very busy analyzing the flowers. So we have a couple questions. Objective A is to compare if there is a positive effect of biopollen which is a wet pollen application. The technology comes from Chile. It's being deployed by some growers here in Ventura County, and they have a big presence in almonds up in the San Joaquin Valley. Then dusting is a dry pollen application where the pollen is blown onto the tree, and then we have the control where it's just whatever happens, happens. This year we're adding a fourth treatment by a company from Israel called Bloom X, and they have a different method. They use electrostatic, so they bring paddles in, and the paddles, they rub up against the inflorescences and by electrostatic charge. So we're adding that treatment this year. The other one is to track the BBCH methodology, is just tracking what stage of flowering a tree is in. The third one is to collect pollen and look at viability and pollen germination. And we have that data from 2025, and it's still being processed. And then the final one that we're collecting is fruit set and estimates of fruit production. So he did estimates, but we still have to collect the yield data. And we went out in the field yesterday looking at the field, the different sites, looking at the yield. But of course, we have 50 trees per treatment at each of the sites. And so we're waiting to get the harvest data. And then finally, we're going to do paternity tests so that we can estimate the effectiveness of these different pollen application methods on fruit set and yield and how they contribute to the yield. So those are the objectives of the project, and we're waiting to get the yield data from last year's research because none of the blocks have been harvested yet. So he looked at the pollen tube germination and the pollen tube growth down into the ovule, and because he's a data guy, I'm not a statistician, but normally when you're analyzing data, you assume that you have a normal distribution of data. But as Anaki showed with pollen, a number of pollen grains on the sigma and the growth, et cetera, you have more of an abnormal distribution. So some of us who are not really great statisticians, I might still do an ANOVA analysis, which is incorrect. But if you have this uneven distribution of data, then you should do all these fancy things that he has on the right-hand side of the slide, which is what he's doing right now. And see if I can go back if I forgot. Okay. So his point here is that it's very you should not be looking at the averages, because in sample one, you can see there's a lot of variability. You divide that up, and you get an average of 20. And in sample number 2, which is what our data is, we have lots and lots of zeros, just like what Anaki showed about the number of pollen grains on the sigma. Well, the data here in California is the same. We have lots and lots and lots of flowers, no matter what the treatment is, with no pollen grains. Okay, and so, but you still get the same average. So how do you interpret that? That's his point. And so he has been working actually with another researcher in the Department of Computing Science, and I can't ever, Evangelos Papalekis, and we've been working with that. Now, another example he has for this to sort of illustrate this is in the lower right. If half of your body is at 14 degrees Fahrenheit and the other half is at 180, the average temperature of your body would be 96.8. But obviously, you can't be 14 degrees or 180 degrees, because if you did that, you would be dead, okay? So even though the average is right, is biologically doesn't make sense. So and then also in looking at correlations with data that's not evenly distributed, an example would be in a city, if you have an increase in the number of potholes, may be strongly correlated with mortality rates, but that doesn't mean the pothole causes deaths. And that's similar with the data that Hamatal just showed. You know, we can show that we have a lot of alternative insects visiting the avocado flower, but until she collects the data saying, yes, you know, these are definitely involved in pollination, it's only a correlation. It's not causation. Okay, another example is that trees receiving supplemental pollen may show a higher fruit set. And so you say, okay, there's a strong correlation between pollen application and fruit production. But that doesn't mean that that added pollen caused the increase. So we have to prove that. And that's what the paternity test is going to be doing, because we are, in many instances, they have Zutano pollinizers in the field or nearby. So then we're applying bacon pollen. And we have one site so that we can say, okay, if we do the paternity test and the paternal source of the pollen was bacon, then we know that the pollen application method worked. So that is the end of the first presentation on this project. It's being funded by the California Avocado Commission. I'm sorry to say that we don't have the yield data yet, so we're hesitant to make any statements about the efficacy of the different treatments. And we're quite excited to be adding the Bloom X treatment, which is the electrostatic one, because preliminary data both in South Africa and Mexico would suggest that that one is an effective method, but we won't know until we test it here. So maybe I'll go on to the next presentation. They're going to switch. Great. Okay, so this project that I'm going to briefly talk to you about, this is another interest of Marllon. This project is being funded by Eurosemias, which is funding the avocado sign breeding program. And it was an outgrowth of the sign breeding program, because we need to know the varieties that we're releasing, actually why they set fruit, especially for Luna UCR. The other funding source we have for this is from the Hansen Trust. So we received a Hansen Trust grant to support this project as well. So this is part of Marllon's work at UC Riverside. I won't go through this, but basically you have the male and the female stages of flowers, as Anaki talked about. And there's 4 hypotheses in this little project. Is climate change impacting the behavior of the avocado flower? And Anaki showed us some data that yes, perhaps yes. Is climate change affecting the pollination time by the honeybee in the avocado trees? So that's possible. Is climate changing affecting the productivity of the avocado tree? Yeah, that's possible. And this is more, goes back into the breeding program, which cultivar in the future is going to be more efficient in producing fruit or being a pollen donor. So because Marllon has his background in big data management and actually machine learning, we've teamed up with Evangelos over in the Department of Computing Science and Engineering at UC Riverside, and we've been working on these questions. So Anaki alluded to this. There's quite a few papers out there showing that temperature plays a role in at least the opening of the female phase of the flower. Not less is known about the male flower. And in that BARD project I alluded to that we had with Sharoni Shafir and Unknown Dog, Loretta Bates, who was working with me at Derbyshire, her data indicated that it was the minimum, pre-dawn minimum, that actually could predict when the female flower was going to open. So, but everything is based on temperature. And so the idea here was to look at flowering at various places in California and elsewhere that have different climate regimes to see if we could build a model to predict when the flower will be open. So we have a site at Lynn Cove up in the San Joaquin Valley, where we're going to have a meeting again on March 12th, if you're interested. We collected data at Irvine. We collected data at UC Riverside, where we have avocados. We collected data in Peru, And we collected data in Brazil. So Peru and Brazil was so that we had very different environments compared to California. And so in the California data set we have was actually Marllon going out and taking flowers and visiting the flowers every few hours and recording what they were doing, day or night. But when we went down to Peru and Brazil, we bought cameras and we put the cameras up so that we could record what the flowers were doing. And so this is the work that we're doing in collaboration with the Department of Computer Science at Riverside. The first question is, can we develop an algorithm that can look at these videos and predict if the flower is open or closed? And currently, with the current iteration of the algorithm that they have, the model is 80% correct in predicting whether the flower would be open or closed. So that's pretty good. We need to improve it, but that's pretty good. The next question is it male or female? So we don't have good efficiency in predicting whether it would be male, but on the female, it's 90% accurate in predicting that the flower, just by looking at a little video, it can predict whether it's going to be male or female. And I'm not sure what these pictures are below. He would have been able to explain what that is, but this is something that he's working on at UC Riverside. So the algorithm is able to identify whether the flower is at different stages. He believes, we can put cameras out, and with machine learning, we can go through these tetrabytes of data, videos he has, to predict if the flower is open or closed, whether it's male or female, and ultimately, when you look at the male stage, is it not just opening as male, but is the pollen being released? And that is the more difficult question, and that's why currently the model is not so good for the male phase. So these are just videos again. Follow the flowers that are in the red circle. So we're going to look at the one at the left. Initially, you can see the flower is opening at female. And when it's bright like this, that means the flower was open at night. So you can see in this case, this is Fuerte in Peru. And the female flower was open from 5 to 1:30 in the morning because it was cold. It was delayed opening. What? What do you mean, how many sites? No, this is multiple sites. This is just one inflorescence at one site. Okay, now if we look at the right-hand side, here the female, this is Fuerte again, open from 4 to 11:30 at night. So when it's a bright light, that's a flash from the camera. We're taking pictures about every 15, 20 minutes, and you can see the flower is closing. Okay. And then this is what the algorithm is looking at. So here's the male phase. If we look on the left, here, this is Jim in Santa Paula. So this data was collected last year, actually. He had the cameras out here in Ventura County. I should recognize the fact of Camille and her research team at Brokaw Nursery has also been really helping with this little research project. So here you can see in the red circle, you see a female flower. We open that. The flower is closing. You can see the male flower there. So there wasn't quite any overlap there. That flower is closing. So the male was open from 12 p.m. to about 8 o'clock at night. And if we look on the right-hand side, this is where the gem, it was a cold day, and the bright light again, this flower is open at night. So it's not likely that unless we had a moth or something visiting, that the honeybee would ever get the pollen from that one. So you can't read this slide, but he's collecting 63 different parameters related to flowering in all this data. And we have temperature monitors in there, humidity, light, etc. And so, again, he's into statistics. He has 63 parameters. How do you decide which one is the most important? And we have a model that can predict when the thing is going to be the tree is going to be in the female phase. And they're working on it. The goal is to have an accurate prediction. And so we're not going to go into this slide because the bottom line, the most important thing is the thing in the bottom left. And the point on here is that previous predictions have relied on temperature, and it's always revolved around, for the female opening, around how warm or cold it was the previous day before the female flower opens. But the data he's collected from all these multiple sites around the world from multiple varieties, it indicates that relative humidity is much more important. It's contributing more to the opening of the female phase than temperature, although temperature is the second most important item. So here's average relative humidity the previous day before the flower opens, and here's temperature, minimum temperature the previous day. So this summarizes it, that these are the most, this is sort of the order or the hierarchy of the factors. And so he's still working on this, and we're still working on it with, but you can see there's solar radiation does play a role. It's the third most important factor. Dew points related to relative humidity. But we still have to figure out the male phase. So we're still working on that. And there was a question earlier about solar radiation. You can see solar radiation is down, but ETO is important, et cetera, et cetera. So the idea of developing a model that would be available to California avocado growers and growers around the world is sort of to predict if you're planting new avocados in a new area, perhaps Hass is not going to be the ideal variety to grow in there. Or if you're looking for pollinizer varieties, maybe we can look at the data and predict what would be a more reliable pollinator variety, a pollinizer variety for another A-type flower. So that is sort of the goal of what he wants to do here. And he's really driven by this particular project. So with that, I'd like to thank you.
In the model you're talking about, is the model interpreting the video, or is the model able to look at general atmospheric conditions? Because I couldn't quite understand. It sounded
Okay,
like
so the model's interpreting the video in light of atmospheric conditions. So we're collecting both of them simultaneously in each location.
But ultimately, we won't need the video if we've understood from his analysis.
Exactly, exactly. So the idea is, if you could say, okay, if the trend is this, then what varieties are adapted to these conditions? Or let's say you're trying to time a spray and the trees are still flowering. You could look at this data saying, okay, on this particular day for this particular variety, when will the female flowers be open? Or if some of these supplemental pollen applications work, you could use that data to say, okay, when is the best time to go out with my supplemental pollen application?
Yes, I remember several years ago, there was a new product on the market, Gly something or other, that was supposed to help in pollination. Is there any new records on what's going on with that compared with what you're doing here?
I'm not sure what you're talking about, so I can't comment on that. When we set up this project, we went and asked people, and there are growers here in Ventura using biopollen products, and there are growers dusting dry pollen. So that's why we have those two treatments. And based on visiting South Africa and also interacting with Alejandro Barrientos in Mexico, who's a very good researcher, people there, both in Mexico and South Africa, they're very excited about this BloomX application method. So that's why we've added them, and we're very happy that they've agreed to work with us here.
Mary Lou, do you see any application? Some of us are doing the jib applications as one of our bloom sprays. Do you see this helping tighten up that?
That's a great question, Derek. Yes, I think, because we all know the avocados bloom normally for many, many weeks. So when is the best time? And I think this might help with it. We'll have to work with Carol. Maybe we could do different, it'll take a little bit of effort, but you're right. I think that would help.